Dear students, welcome to the next lecture on colloids. In the previous lecture, we have discussed about the preparation of colloids. In this lecture, we will discuss about the purification of colloids prepared. The presence of impurities, particularly electrolytes, can make the salts unstable. And these impurities must be eliminated by suitable means. The methods for purification of salts include dialysis, electrodialysis, and ultrafiltration. First one is dialysis. The particles in a true solution can easily diffuse through a parchment membrane or other fine membranes, whereas colloidal particles, being much larger, cannot be diffused. If a mixture containing colloidal particles and a true solution is placed in a parchment bag, which is then held in a wide vessel containing pure water, the substance in the true solution pass out while the colloids remain in the bag. The figure represents the dialysis process. So in the parchment bag, there is the prepared colloid and the true solution, and it is placed in a big vessel containing water. The water is replaced frequently. There is an inlet and an outlet, and during the dialysis process, the particles of the true solution will pass through the membrane and it is washed out by water while the salt particles remain in the parchment bag. Thus, dialysis can be defined as the process of separating substances in a colloidal state from those present in a true solution with the help of fine membranes, and the membrane used for the dialysis purpose is called dialyzer. Usually the process of dialysis is quite slow, but that can be quickened by applying an electric field if the substance in the true solution is an electrolyte. When an electric field is applied, the process is called electrodialysis. The mixture is placed between two dialysing membranes and pure water is taken in a compartment on each side. Each compartment contains one electrode. One is the anode and the other is the cathode. And the required voltage is applied to the system. The ions of the electrolyte migrate out to the oppositely charged electrodes and the colloids remain in the parchment membrane. So electrodialysis is very similar to dialysis, but in the case of electrodialysis, electric field is applied by means of two electrodes, and this makes the dialysis process very fast. Next method for the purification of salts is ultrafiltration. Ultrafiltration is the process of removing soluble impurities from a colloidal system by allowing them to diffuse through an ultrafilter. An ultrafilter is a specially prepared graded filter paper through which solvent molecules and the impurity ions can pass out, but not colloidal particles. Because an ultrafilter is one having the pore size which is sufficiently small to retain the colloidal particles. One of the materials used for the preparation of ultrafilter is cellophane. Also, ultrafilters can be prepared from ordinary filter papers by making their pore size small enough to prevent the passage of colloidal particles. The pores of the filter paper can be made smaller by treating it with a solution of gelatin and subsequently hardening them by soaking in formaldehyde. The ultra filter is fitted using supporting wire frames on a vessel. The impure sol is then poured over the filter. The impurity solution diffuses through the filter and pure sol is retained. Since the normal process is very slow, pressure or suction is usually used to force the impurity solution to come out of the sol through the filter, and thus applying pressure can fasten the process of ultrafiltration.